Great point. I guess the final question I have for you in your work, where do you think the future of genetics and epigenetics, DNA, CRISPR, like, is there anything that you're seeing now, since you're in that world, that is kind of going to be emerging, or maybe some of the threats, just something to look into?
I think there are real positives, and I think the real positives are to do with things like treatment of human genetic diseases at much lower cost and with much better outcomes for patients. I think CRISPR is going to be an absolute game changer on that. I think we'll also see it building more robustness into our agricultural systems, because we can actually create specific varieties of plant crops that will respond to particular stimuli or threats, but without breeding out all the other potentially useful bits in those crops. I think those are incredibly important. I think the immediate short-term downsides of things like CRISPR is we will start seeing unethical practice in terms of poorly regulated areas, where we'll see things like gene editing tourism, where prospective parents will be basically sold a lie about, we can do these changes to your offspring while they're still tiny embryos, and they will have a better chance of being clever or tall or whatever. We will start seeing real snake oil applications of CRISPR, or even just people claiming they're using CRISPR and they're not. They're basically just conning prospective parents. Right. I think that one's going to be very interesting to watch. I think the ones that are going to be potentially really scary with CRISPR and gene editing are if we start interfering at an ecosystem level by, say, introducing lethal mutations into insect populations that will spread through those insect populations, and we don't understand the ecological consequences of that. And also the fact you can start mucking around with pathogens really easily using CRISPR. Really, really nervous about that when you see people going, hey, look at this highly toxic strain we've been able to create in 3 weeks in the lab. That seems to me the sort of thing where we would need a lot more think now, act later, rather than the other way around.
Gotcha. There even was something that came out recently that Boston University has created a new strain of COVID. I don't know if you saw that. And I think the mortality rate on that strain in mice is like 80%. Like, it's the most insane thing. I don't understand why we do it. And also it gives so much fuel to the people that are saying this was,
I think there have
the
to be
origin of it,
much stronger justifications and pre-experimental regulation before we let people be doing that, rather than them going, oh, I've done that. Was that a good idea? I don't know. Maybe it wasn't the best idea. That's not helpful in the current situation.
No. Like, fine, give meth to mice. Stop messing with viruses.
Absolutely,
like, have we not learned anything? Like,
exactly.
Well, I think we know the answer to that. We really haven't.
No, we probably haven't.
Right. Well, Nessa, thanks again. It's lovely seeing you again, and always a pleasure to chat with you.
You too, Michael. It's been lovely. Thanks a lot. Bye.
Bye.